we're going to talk about a whole bunch of things regarding midterm 2 today. And so first of all, this is on Friday. And so it's going to be in class, on paper, no matter what's going on with the IT issues. You need to be on campus on Friday for this midterm. So that's at 4 o'clock, 5 o'clock in the usual place, S210 on campus. This is what the midterm is going to look like. It's going to have some multiple choice true false. It's going to have a mitosis meiosis question. We did a sample one last day, and if we get to it, we'll do a sample today. There's going to be some short answer questions. I'll show you what they look like. I have some samples here in this review. Matching, we've done that before, and the long answer is going to be a metabolic pathway question that I told you a little bit about already. And I have a sample in here as well. I think it's the same sample I showed you last day. We are going to cover in the midterm topics number 7 all the way through topic 12. And so what you want to do is study those ones. Don't worry about 1 to 6. Start at the cytoskeleton and work your way all through to the cell cycle. So these last 3 topics here, so respiration, photosynthesis, and the cell cycle, those are the ones that we spent the most amount of time on. And all the other 3 were like 1 lecture each. So I would, study twice as much for respiration as you do for enzymes and so on. Okay, so what I want to do is just kind of overview some of the things that we talked about today. And I have a few questions to show you, sample questions, and a few notes to make as we go along. And so we'll do all that. So way back to topic 7, we were talking about the cytoskeleton. And we made a table, and we talked about the different functions of the different components of the cytoskeleton. So you need to know that, right? What these things are, what they're made of, and what they do. And so I've kind of summarized that here on this slide, and I'm just going to quickly talk about them for a minute or two. So microtubules, these are the largest ones, and they're made out of tubulin. So that's a type of protein. All these are proteins, by the way. That's kind of the definition of the cytoskeleton, is that it's proteinaceous fibers made out of proteins. So tubulin forms microtubules, which are giant tubes. And this is used for a whole bunch of different things, but kind of the main things that we talked about were cilia and flagella, the eukaryote version, of course. Spindle fibers, we just talked about in topic 12. And also organelle movement, so moving vesicles and other organelles around the cell. The smallest ones are the microfilaments. These are also called actin filaments. Actin filaments. And these are kind of like a rope. You can see in the image there, you've got these actin proteins and kind of like 2 little strands wrapped around each other, making a very, very skinny kind of fiber. So these have a lot of specialized functions. We talked about muscle contraction as actin and myosin. We talked about amoeboid movement, formation of the cleavage furrow in topic 12 with cell division. We also talked about microvilli. So microvilli are those intestinal cells that kind of, they look something like this, right? And that whole idea is to increase the surface area for absorption during your digestive processes. And the last one here is the intermediate filaments. Also tend to be quite specialized in different types of tissues. The main one we talked about way back in topic 6, we talked about the nuclear lamina. And one other example you should know, of course, are the microvilli, which are the same microvilli that are built of actin filaments. The intermediate filaments are part of that actual structure as well. So intermediate filaments, if you don't get it from the name, they are intermediate in size. So hopefully that helps you remember which are which. So you're going to need to know a little bit about all of these. And one thing that we did focus on a little bit, and we also, I believe, had a question in our lab report on this, the lab 4 or 5 lab report on comparing the eukaryotic versus the bacterial flagella. And so I would expect you to know a little bit about some of the differences there. In the past, this has been a long answer type of question. It won't be a long answer type of question, but it'll probably be somewhere else on the midterm. So I would expect you to learn a little bit about these. And you can see some of the features are compared there. Eukaryotic flagella is bigger. Eukaryotes are bigger in general. It's made out of microtubules. It's surrounded by a membrane. It's powered by motor proteins such as dynein, which use ATP. And it kind of flip-flops back and forth like a big tail or a whip. Whereas the bacterial flagellum is powered by the proton motive force. It's made of flagellin protein, and it moves in a rotating kind of manner, clockwise and counterclockwise. So here's a sample multiple choice question. So it says your intermediate filaments are assembled from actin monomers. No, that would be microfilaments. Play a major role in the contraction of skeletal muscle. No, that is also actin. Number 3 are permanent cell structures broken down during cell division. So that would be the nuclear lamina. And that is correct. What about the other ones? Guidement of movement of organelles, that would be microtubules. So MT for microtubule are the largest of the cytoskeletal structures. That would be the microtubules as well. So number C is the correct answer. All right, topic 8 is cellular order and energetics. I know I'm going a little fast. I want to slow down when I get to respiration and photosynthesis, by the way. So here's a little cartoon for you. You can see he says he's got a bottle of beer next to a mug of coffee, and he says the beer is getting warmer and the coffee is getting colder, and he's not too happy about that. And he says, someday the opposite will happen. On that day, I will mock the laws of thermodynamics. And of course, he says not today. So it's kind of a little joke there. But remember, we talked about energy and thermodynamics and a whole bunch of other definitions. So I want you to go back to this unit and take a look at some of the definitions that we went over. And we finished off this unit talking about ATP and cellular work. Remember, cellular work involves movement of things, so movement of organelles, movement of things across membranes, and lastly, it involves activation of chemicals, which we called energy coupling. Energy coupling. And there's a diagram from the textbook showing the concept of energy coupling. This is the synthesis of glutamine, an amino acid, which is formed by 2 parts. Glutamic acid and ammonia, and you can see the delta G on this reaction is too great. It's a positive delta G, which means that it will not proceed naturally without the input of energy. So where do we get that energy from? We get that energy from ATP. And so you can see that this becomes a 2-step reaction where ATP is added, and then that energizes or adds the activation energy for the second reaction, which is the addition of the NH3 functional group. And overall, the net reaction is negative, which means it is a spontaneous favorable reaction that can proceed thermodynamically. All right, here's a sample question. Here we have to think about the definitions again. This is why I keep saying, hey, you've got to go over all the words that we learned and understand what they mean so that you can use them in a sentence. So this one says, during an endergonic reaction. So remember that ender means in or within, and gonic means energy. So that means you're bringing energy in, okay, not releasing energy. So energy is generated, that usually means energy is being released. So that one's a no. Potential energy is increased. So that's kind of like the little guy. Here's my little guy. And the little guy is climbing up the ladder, and then remember they're about to jump in the lake. So that is gaining energy, so that is correct. Let's just take a look at the other ones here for a moment though, just to make sure that that makes sense. Entropy is increasing. Entropy is disorder. So in this case here, the guy is taking energy and is actually getting more order, so that one is incorrect. Order is decreased, nope. Energy is released, so none of those seem to make sense. So the correct answer is definitely B. Potential energy is increased. Okay, so of course that means not necessarily the guy on the diving board. It means potential energy is increased within chemical bonds. Topic 9, enzymes. So enzymes, we talked about activation energy, we talked about active sites, and again, a bunch of kind of lower end concepts. And so make sure you go over this unit and take a look at what we covered. Hopefully that information can be used to help us with respiration and photosynthesis. And we did talk about phosphofructokinase. I think maybe we talked about that in either this unit or the glycolysis unit as being the key regulatory enzyme. So make sure you know a little bit about phosphofructokinase. So I mentioned there's going to be some short answer questions. Some of the short answer questions might be definition type questions. So here's an example of a definition type question. You can see it says define active site. So remember, when you're answering questions on like tests, 1 mark means roughly 1 long sentence. So it could be 2 sentences. You don't have to be in sentence form on a test, but it means that you're having at least a couple of things in there. And so usually these kind of questions, I make them worth 1 mark, and I think, okay, half a mark for this and half a mark for that. So in this case, it says here, the region of an enzyme that binds a substrate. And that's not just binding the substrate, but there's also catalysis occurring there. Some of these questions, you can throw in examples that will help you with your answer, but make sure that you do kind of have a complete thought, not just something simple. All right, topic 10. We're going to slow down a little bit and talk a little bit about respiration. I think I have, there's our mitochondria there. So just going to quickly label that. Remember, we have an outer membrane, so an OM, and we have an inner membrane, so IM, and this is the intermembrane space. So I've got to write small there to squeeze that in. I apologize for that. And we have the matrix here. And then, of course, this is the inner membrane as well, which sometimes we call the cristae. So in terms of what's important, the matrix, this is where we have the citric acid cycle. And the intermembrane space, this is where we have our proton motive force. So I'm just going to draw it as a bunch of protons. Okay, so let's talk about respiration here. Remember, I told you I want you to think about these 3 things and understanding really what the input and output for every pathway is. Okay, and focusing on these things, right? Don't worry about water and a couple of other irrelevant things. So for electrons, don't forget that means we mean electron carriers. So electron carriers include NAD plus, NADH. Also includes FAD, FADH2, and for photosynthesis includes NADP plus and NADPH. Okay, so that's what we're talking about with electrons. The electrons, of course, are not free electrons. They're stored within chemical bonds. And that's what we care about, is the chemical bonds. So let's take a look and think about these pathways for a few minutes here. And I'm going to make some notes as we go along. So there's kind of the overview of respiration, right? And so I want you to start here and then pick apart each of these things piece by piece and learn some of the key concepts about it. So let's talk about glycolysis. So glycolysis, you can see, starts out with glucose, and our product is 2-pyruvate. So remember that glucose is 6 carbons and pyruvate is 3 carbons. I'm not going to write that down. What else is going on in glycolysis? There's some water as one of the products, but we don't care about water. We care about ATP and we care about those electron carriers. So remember that we are making 2 ATP overall. So in order to make ATP, it's going to cost you 2 there. Maybe I'll just put it on the next line. It's going to take 2 ADP plus 2 inorganic phosphate. That's what the PI is. And there's my 2 ATP. Now, in terms of the details, remember that it actually costs us 2 ATP to do glycolysis, and we make 4 overall. So this 2 that I have here, 2 overall, 2 is the net ATP. Even though we make 4, subtract the 2 that were required to start off the reaction, and you're going to get 2 as your net ATP. So electron carriers, we have 2 NAD+. And as I've said before, don't worry too much about the stoichiometry. You don't have to worry about the 2s. Just know that we're making NADH here. And in order to make NADH, it means we need NAD+. So there's my NADH. And notice, I'm just going to put it as a line there, is that the electrons are in bonds. I'm just putting that as kind of a little note around the NADH. So that's glycolysis. Where does glycolysis take place? Way out in the cytoplasm. And that's why when you look at this diagram, you'll see that they're showing a mitochondria around it. I'm just going to go back a page for a second. You can see that it's showing all of the other steps happen in the mitochondria, and the glycolysis is out there in the cytoplasm. So there's glycolysis. The other thing to remember about glycolysis, remember that phosphofructokinase is the key regulatory enzyme, okay? So don't forget that. All right, step 2 is pyruvate oxidation, okay? So pyruvate oxidation, we start off with pyruvate. So hopefully that is obvious that if we're gonna oxidize something, it better be in the title. So pyruvate oxidation, and we're turning pyruvate into acetyl-CoA. Okay, so what else do we need to know about this? To make acetyl-CoA, we need coenzyme A. So coenzyme A, we write as co-ash. Sometimes you see in textbooks writing it as CoA, but the SH is reminding us that the sulfur hydrogen is an important part of that coenzyme. Sorry about that, I'm just trying to draw my C's a little nicer. There we go. We also lose some CO2 as part of this reaction. And so pyruvate is a 3-carbon compound. Acetyl-CoA has many carbons, but the 2 that we care about at the end, the acetyl group, is 2 carbons. And so where does the extra carbon go? It becomes carbon dioxide. The other thing we could say, we could multiply all these things by 2, because remember in glycolysis we have 2 pyruvate, but we're not going to worry about the stoichiometry. So there's no ATP produced, but we do produce some NADH, 2 overall for every glucose and 1 for every pyruvate. So I'm going to put that one down. The NAD plus becomes NADH. There we go. So there's my reactants and my products. And that's pyruvate oxidation. There's actually a lot going on there, but it's really just one reaction in terms of you're starting off with pyruvate and ending up with acetyl-CoA. All right, so next is the citric acid cycle. So we're going to start off with our acetyl-CoA. So I'm going to stick that in there, acetyl-CoA. Acetyl-CoA. And what does the acetyl-CoA become? Ultimately, those carbons become carbon dioxide. We're going to lose our acetyl group. So there's my coenzyme A. And that's the carbon part. So remember, the other things we care about are the ATP and the electron carriers. So for the electron carriers, we are going to, we're going to make some NADHs. We're going to make 6 overall. But like I said, I'm not going to worry too much about the stoichiometry here. I'm just going to add my NAD plus becomes my NADH. We have that other electron carrier, don't forget, the one called FAD. And it gets reduced and becomes FADH2. And all right, and then our ATP. And so in order to make ATP, we gotta take ADP and inorganic phosphate, and we're gonna make some ATP. So that is the citric acid cycle in a very, very, very simplified context. Now, you know, there's details you could remember. For example, the citric acid cycle actually makes GTP, but that GTP can be easily converted to ATP. They're energetically equivalent. So for all intents and purposes, we've made 1 ATP equivalent. Okay. Other details, you know, if you want to remember the stoichiometry, we're actually making 6 NADH here and 2 FADH2, for example, and those kind of things. And so each of those is, of course, going to get converted into ATP in the last final step. So let's get to that last final step, which is the oxidative phosphorylation. So notice we're taking these NADHs from glycolysis and the electron carriers from the citric acid cycle. And we're going to bring them over here. We're going to bring them to the electron transport chain. And we're going to do some chemiosmosis, which is the production of ATP. So if you want to kind of think about what is going on here, we can look at it this way, right? We have these electron carriers. So the NADHs and the FADH2s, those are kind of the ingredients of oxidative phosphorylation. And of course, they're going to get oxidized. That's the oxidative part. And they're going to get recycled back to their original precursors, right? NAD plus and FAD. Oxidative suggests oxygen, so let's not forget that. So we can add our oxygen in. So notice oxygen is sort of that funny last thing that it's not an electron carrier or ATP or anything like that. But it is ultimately accepting electrons, because remember what it's becoming. It's becoming water in the end. So this is the one time we sort of care about water, because it is accepting, it's part of that electron accepting process. Okay. So what else is going on here? We're not just oxidizing this stuff for the fun of it. We are making ATP. So don't forget to make ATP, what the ingredients are. It's ADP and inorganic phosphate. And ultimately, we're making our ATP. So that's kind of a formula for oxidative phosphorylation. And, it's not balanced here, of course. I'm just showing you what the essentials are. So the ingredients are those electron carriers. They get oxidized. Ultimately, the electrons end up on oxygen and become water. And the energy for this is it's being used to make I guess you could say it's being used to make a proton motive force or an ion gradient, whichever way you want to describe it. And now you end up with a whole boatload of ATP. So that's good. We have our pathways there. So all these things have a few more details around them, but when you start your studying, this is where I want you to start thinking about what is going on in these pathways, what is going in and what is coming out, because ultimately all these things are very important. Here we are back to the kind of summary of everything here. And I did a little ATP accounting for you. And I think the final answer was we make about 30, I can't remember, it's 34 or 36 ATP. And most of those being produced, of course, in the oxidative phosphorylation step. 2 being made in glycolysis and 2 being made in the citric acid cycle. So that's not quite everything. That was almost everything around respiration. The last thing about respiration is to think about what if there is no oxygen? And so I'm just going to go back a slide for a second. So if there's no oxygen, all of these things here shut down. Now, technically, oxidative phosphorylation is the only thing that requires oxygen. But when it shuts down, it actually shuts down everything in the mitochondria. So glycolysis is really the only thing that can occur if there's no oxygen available. And so that is what this slide is showing, is that when the glucose is converted to pyruvate, the fate of that compound really depends on whether there's oxygen or not. If there is oxygen, we go into the mitochondria, we do that pyruvate oxidation, the citric acid cycle, the oxidative phosphorylation, and we make tons and tons of ATP. If we don't, then depending on what organism you are, you might make ethanol or lactate. And so make sure you know a little bit about that as well, okay? Why are we doing this? I think I just explained why, but you may have to look that up, because that will definitely end up on the test there somewhere in terms of what's going on with the ethanol or the lactate. So remember, of course, if you're doing the no oxygen route, you're not making a lot of ATP, but you're making at least some ATP. And that's good enough for yeast and good enough for our muscles, at least in the very short term. So here's a short answer question. So this is one that can be a lot more complex. There's kind of not necessarily different answers to this, but some answers are going to be more right than others. Why do our muscle cells sometimes make lactic acid? Okay, let's just go back to this slide for a second. So this part here, right? No oxygen. That's going to be half a mark for saying something about oxygen is not present. I'll show you what a kind of a detailed answer looks like from a chemistry standpoint. But you know, your answer may or may not necessarily look like this, right? It says when there's not enough oxygen, pyruvate is used as an electron acceptor to regenerate NAD+, and the byproduct is lactic acid. So that's technically what's going on. Your answer, you may get a full point, but not necessarily answer that. Some people may say when there's not enough oxygen, pyruvate is not degraded completely to carbon dioxide. It's only degraded partially to lactic acid. I would accept that. And probably a few other things. But for this kind of one here, you have to have this part here for at least half a mark, because that's the whole point of fermentation. There's not enough oxygen to do the more efficient process. And that's why we do it. Here's a sample long answer question. Like I said, your long answer question is going to be a metabolic pathway, but just showing you what some long answer questions have looked like in the past. And I just want to give you some tips for answering long answer questions, right? So 5 marks. So make sure that you are giving, you know, enough content for me to give you 5 marks. For example, you know, 5 long sentences, I guess you could think of it as. I would say 5 marks is about minimum of half a page. Again, it kind of depends on what the questions are. In this case here, it's asking you for a diagram. And if I ask you for a diagram, don't just draw the diagram and don't explain it. Explain the diagram, what is going on. What, in this case here, the differences between the energy sources and those type of things. Okay, let's talk a little bit about photosynthesis, and then I have an exercise where I want to compare respiration and photosynthesis, the one that I had alluded to in class. Very quickly, I will label this chloroplast. Remember, we have an outer membrane, and we have an inner membrane. Number 2 is the intermembrane space. It's not even important because nothing important goes on in there. But what is important are the number 5, which is the thylakoids. And this here is the stroma. And inside the thylakoids is the thylakoid space. So I apologize for the lack of room there. I was not anticipating having to do this on PowerPoint or on Zoom. I mean, I was anticipating doing this on a whiteboard. All right, let's talk about the major steps of photosynthesis. So don't forget this slide, okay? This summarizes everything here. So what I want to do is kind of pick this apart again, like I did for the last one. So there are the light reactions, okay? And I actually have another slide I'm going to show you that's outlining the light reactions. So I'm going to get right to that. And this is showing the light reactions with just a little bit more detail in terms of the photosystems and whatnot. But again, you can kind of write this as a formula in terms of what is going on in the light reactions, right? So the light reactions, of course, are going to involve light. So don't forget light. They're also going to involve water. And I'm just looking at the diagram here and making sure I have everything that is going to be included in there. We're also going to, so what I'll do is I'll put the arrow over here like this. So light doesn't become something, it ends up in chemical bonds. The water ultimately becomes oxygen. And if you take a look here, we're making some ATP over here, and we're making some NADPH. So let's throw those things in here. So we've got the ATP. In order to make ATP, I need ADP and inorganic phosphate. And to make ADP, or sorry, to make NADPH, I need NADP plus. And so there's my NADPH there. And that's the light reactions, okay? You can get into the details and think about the chlorophyll a little bit. You can talk about the proton motive force here and those kind of things. So make sure you go in and take a look at some of the definitions and kind of details around that. You can see that this is done right on the thylakoids. So You could label this as the thylakoid membrane right there. And of course, the chlorophyll is in these green things, which are often called photosystems or reaction centers. They have a whole bunch of different names. But those are the light reactions. And you take a look way back at this, it's all the same ingredients, right? We have light, check. We have water, check. NADP plus, ADP, and then our final products are oxygen, ATP, and NADPH. And that's everything that I have right there. Kelvin cycle. So what's going on the Kelvin cycle? So Kelvin cycle, we have the things that we made in the light reactions. So in the light reactions, remember we made ATP and we made NADPH. Not the oxygen though. Remember the oxygen is of course, it's just a byproduct. It's going away, right? So ATP plus NADPH. And then of course we have up here, we have the CO2. Okay, so we're going to take that and we're going to break down ATP into ADP plus inorganic phosphate. We're going to break down the NADPH into NADP plus, and we're going to make some sugar, and the sugar is G3P. Remember that G3P is glyceraldehyde, 3-phosphate, and this is used to make other sugars. And what other sugars are we making? Often glucose. Glucose is often the default sugar, but it could be used to make other things: fructose, galactose, maltose, starch, etc., etc., etc. And it just depends on what the plant is wanting to do with its sugar. But glyceraldehyde 3-phosphate is technically the final product. Again, notice no stoichiometry here. I can never remember the numbers myself for the Kelvin cycle, but it's a lot of ATP and a lot of NADPH are used to take that carbon out of the atmosphere and fix it. So sometimes the Kelvin cycle, by the way, remember, is called carbon fixation. Carbon fixation. And remember, the big enzyme up there is Rubisco. That's the important enzyme, Rubisco. My C's are not very good today. Okay, so what I really wanted to get to today was comparing respiration and photosynthesis. But that was kind of the big summary of photosynthesis there. Here's a sample multiple choice question. And so again, we have to know our definitions. And what is chemiosmosis? Chemiosmosis is ATP synthesis using the proton motive force. So let's take a look. Creation of a proton gradient, correct? Rubisco, what is going on with Rubisco? That's my previous slide. Remember, it happens right here. There's no ATP being produced there at all. So that one is incorrect. Phosphorylation of ADP, that's ATP making ATP. ATP synthase makes ATP. Oxidative phosphorylation makes ATP. So the correct answer here is B. Rubisco has nothing to do with ATP at all. Okay, so don't forget, purpose of the light-dependent reactions, true or false, is used to produce ATP and oxygen. False, that's not the purpose. Oxygen is not the purpose of the light-dependent reactions. The plant at this point does not care about oxygen. We want to make ATP and NADPH. So here's that sample long answer question. I think I showed you this last day. You're gonna have a sample long answer question and I'm gonna ask you a whole bunch of things about it. Some of the questions I'm gonna ask are what is it? Maybe where does it occur? And then I'm gonna ask you a lot about the reactants and the products. So make sure you know what all of these molecules are for. ATP and ADP, they have their uses in different places. NADH and NAD+, oxygen, carbon dioxide, glucose, pyruvate, acetyl-CoA, all those things. For each reaction, you should know what their ultimate purpose is. Sometimes their ultimate purpose is to go to the next pathway, right? If we're looking at the light reactions of photosynthesis, what is the purpose of ATP there? The purpose of ATP with the light reactions is not cellular work. The purpose is to go to the Kelvin cycle and then be used for carbon fixation. So think about those in mind when you are studying these pathways. Okay, so here's what I want to spend a little bit of time on today, and we'll see how much time this takes. So it says compare and contrast electron transport chains and ATP synthesis in mitochondria and chloroplasts. So what I want to do, usually I do this on the whiteboard. So I've got a Word document prepared here, and we're going to compare photosynthesis and respiration with a whole bunch of different metrics, and some very simple, and then some a little bit more complex, just to give you a different way to think a little bit about respiration and photosynthesis. Okay, so first question, it says, where are mitochondria and chloroplasts found? Okay, so mitochondria, where are they found? They're found in all eukaryotes. So what does that mean? It means plants and animals. Okay, mitochondria are not just found in animals. For some reason, some people want to tell me that all the time. Fungi, protists, algae, all of our favorite eukaryotes. Where are chloroplasts found? Chloroplasts are found in eukaryotic autotrophs. So autotroph means self-feeding organism. And so what does that mean? It means plants and some protists. Maybe we'll call them photosynthetic protists. Photosynthetic protists, and I guess we could add algae onto that list as well. Algae, sometimes people consider algae to be a type of protist. Some people group them with plants. I think we're kind of at a stage where we're mostly grouping them in their own category nowadays, in that they are very plant-like and they're definitely related to plants, but they're just not as structurally complex. All right, so question 2, what happens here with mitochondria? So we could say at a very general level, cellular respiration, but really it's only the last 3 stages, which is pyruvate oxidation. Pyruvate oxidation, I can't spell today, there we go. Pyruvate oxidation. The second step is the citric acid cycle. And the third step, of course, is oxidative phosphorylation. So those are all the steps we're talking. Maybe what I'll do is I will do this. Give them each of their own bullet point, easier to maybe read. Okay. And as I said before, if people are missing something here, you can always ask me to email you the Word document or something like that. It's not really a big deal. I'll just save this to my computer. What happens at the chloroplast? Photosynthesis. So remember photosynthesis, we kind of break it into 2 parts. We call it the light reactions or the light dependent reactions. And part 2 is, of course, the Kelvin cycle, which probably better off being called carbon fixation. Okay, so there are other things that go on in mitochondria and chloroplasts. In chloroplasts, we store starch and things like that, but we're really focusing today on respiration versus photosynthesis. Okay, so here are where the more difficult questions come in. It says, where are the energy-rich electrons from? Okay, so let's think about this for a minute. Energy-rich electrons. So remember, I kept emphasizing that electrons are never sitting around loose, okay? They're in chemical bonds. So in respiration, what chemical bonds are we getting energy from? We are getting energy from chemical bonds in glucose or our food, okay? So we can say organic molecules, I guess, and we'll be able to say example glucose, because that is the best fuel source. It's a good fuel source for our brain, and our cells love glucose, although we could be eating other things, right? That's why I put it generally as organic molecules. Okay, so, and if you want, you could put electron bond, chemical bonds in organic molecules, but that's fine. What I'm going to do is I'll get back to chloroplasts here in a moment. I'm just going to answer the next couple of questions here in the mitochondria first, and then we can go back to photosynthesis to compare. So where do the electrons go? So glucose gets broken down into eventually pyruvate into acetyl-CoA into carbon dioxide, so it's getting stripped of electrons. And then remember those electrons, they end up on the electron carriers. Okay, so maybe this is the next best question. What are the electron carriers? The electron carriers are NAD+ / NADH. And the other electron carrier is FAD, FADH2. And what I'll do is put a little subscript on there for you. So subscript on that. And the plus, I will make that a superscript. There we go. Okay, so the electrons get stripped off glucose. Eventually becomes carbon dioxide. They end up on NAD+, become NADH, and they end up on FAD and become FADH2. That goes to the electron transport chain. They zip through the electron transport chain, and the final electron acceptor is oxygen. So O2. And then we can say it gets reduced, meaning it's gaining electrons. It gets reduced to H2O. Okay, so let me just, I'll make those small letters again for you. And that way it looks a little bit nicer. Okay, so that's respiration. We still have a few more questions. We'll get to those in a minute, okay? I think I've got 3 more questions. We'll get to that. That's more about the ATP synthesis part. Photosynthesis. Maybe we'll start off the electron carriers. That's the easy one. Electron carrier in this case is NADP+. Remember, it's very similar to NAD plus, but it has an extra phosphate on it. And that becomes NADPH. Okay, so I will make that plus sign the right. Oopsies, what did I just do? There we go. I'm just going to make the plus sign a superscript. There it is. Uh-oh, what did I do now? I think I want to go back to there. There we go. Okay, so there's our electron carriers. They're all good to go. Okay, now let's talk about these energy-rich electrons and the final electron acceptor. So here's where it's useful to think of photosynthesis kind of being the backwards of respiration. Okay, so go to the beginning of photosynthesis and think about what our ingredients are. We had light, we had ADP and NADP+, so no, those are carrying electrons, they're not the source of them. And the last thing was water. So remember that water gets broken down and it becomes oxygen. So that's ultimately where the electrons come from. And you can see how this is kind of the opposite of respiration, and that respiration produces water. So we can say that water, and it gets oxidized to becomes oxygen. There we go. So not as hard as you think when you know these pathways really well. These questions are not as hard as they look. So where did the electrons go, right? So the electrons, they go on the electron carriers, right? And then where do those electron carriers go? They go over to the Kelvin cycle. And what happens at the Kelvin cycle? The electron carriers are used to reduce CO2. So we can say that is that and what does the CO2 become? The CO2 becomes organic molecules. Right, and of course, the examples are G3P, glucose, and many, many other possible things. Okay. Just if I'm going too fast, just let me know. If anyone has any questions, feel free to jump in. I'm going to scroll up a little bit to see these last few questions. All right, so you can see these 3 questions. We're really just asking about the electrons. Okay, so what I want to do now is think a little bit more about the ATP part, and that's the last 3 questions here, and unfortunately spill over onto the next page. So it says here, where is the proton motive force? Okay, so go back to our mitochondria, and remember we had some different areas in the mitochondria. The innermost section is the matrix, and the matrix is where the citric acid cycle is going on, and the protons are pumped from the matrix, and they're pumped into the intermembrane space, and that's the only other space we have there. Membrane space. Okay, where did they flow to? They flow from the intermembrane space. Like I said, you need to put all these words, membrane space, to the matrix. Okay, and the last question, it says, what is the ATP used for? So why are we making ATP in the mitochondria? We're making it for this thing here that we call cellular work. Okay, and remember that cellular work, some of the examples of cellular work include movement, so movement of organelles or movement of muscles, transport of things across the membrane, so we'll call that membrane transport, and energy coupling is the last one. Of course, there are other, probably other examples of cellular work. I can't think of any off the top of my head at the moment, but those are kind of the three that we discussed in class. So that's respiration, right? The whole point of respiration is this last thing, making ATP and having that ATP used for cellular work, which means we can exist as living organisms and we can build things, we can grow, we can do all the things we love to do as living organisms. All right, 3 more blanks for photosynthesis. Where is the proton motive force? So remember, the light reactions, they take place in the thylakoid membranes, right? So the protons are pumped into the thylakoid space. Okay, and where do the protons flow? They flow out of the thylakoid space and they flow into stroma. So the stroma is the big kind of area in the chloroplast. I kind of think of it as sort of the chloroplast cytoplasm. And that's where the Kelvin cycle is, right? Because we want ATP for the Kelvin cycle. And I guess I just answered that last question. What is the ATP used for? The ATP is used to reduce CO2 in the Kelvin cycle. You can also think of it as carbon fixation. I guess technically that is a type of cellular work. In this case here, it's the energy coupling kind of cellular work. But overall, what the answer I'm looking for in this kind of question is, what is the ATP used for? It's used for the Kelvin cycle, and the Kelvin cycle is used to take CO2, reduce it, and make organic compounds, which is that process called carbon fixation. Okay, so I'm hoping everybody got that. See, there's a couple minutes left. I'm just going to go flip back to the PowerPoint and see what else there was I was going to say today. And because that's kind of near the end, I guess I'm just going to take a quick look through this PowerPoint here. There's cell division. So I had a little bit more to talk about with mitosis and meiosis. And you can see there's another mitosis-meiosis problem I have here. And what I'm going to do is I'm going to leave that till Wednesday. And Wednesday, I'm hoping we're going to be in person. And so bring some paper, and I'm going to get you to do your own mitosis-meiosis problem. And Wednesday's review session is going to be a little bit more interactive in that you are going to be doing some of the work, and I'll be taking a look at it. Hopefully by then, you'll have had a chance to study a bit, and it will be a kind of a different kind of review session. So there's the problem to look at. And anyway, so good luck with your studying. Good luck on the midterm. I will see you Wednesday. There's lots of time to get all this in your head and time to ask questions and things like that. But it looks like we're done with our time today. So thank you for coming, and I will see you next time.